is called the axon terminal or the axonic bulb. There's a synaptic bulb is also the other thing here. So this is the axon terminal, this is a telodendria, and that's an axon. And that term telodendria is what I had forgotten to talk about. So we've spent the majority of this class talking about neurons, the structure of neurons, the function of neurons, how they work generally, and we'll get into greater detail tomorrow and then Monday. But before we do that, we're going to talk about the other helping cells, and those helping cells are called glia or neuroglial cells. Those glia or neuroglial cells come in 6 varieties. There's 2 in the peripheral nervous system, and there's 4 in the central nervous system. This list here shows them all the different types. So starting in the central nervous system, we have 4. You have astrocytes, ependymal cells, microglia, and oligodendrocytes. Along with those names right here on the left-hand side, you can see the generalized function. And generally speaking, when you're talking about function here, there's either protection or support. So oligodendrocytes will protect and insulate an axon. Astrocytes will support a neuron, just in general. And then ependymal cells and microglial cells work together. Ependymal cells produce what's called CSF, or cerebral spinal fluid, and microglia are found within the cerebral spinal fluid, and they are also helping protect. So generally speaking here, when you're talking about support, you're talking about bringing nutrients to the neurons, because these are nutrient hungry cells. They are incredibly oxygen and glucose dependent, and they require it constantly. So they need additional support there. And then remember too, now neurons are not undergoing mitosis and cytokinesis. And so these neurons, I've said this already, that neurons are older than you are, neurons in your body. The neurons in my body are older than I am. If we measure our birthday as the day which we were born, we had neurons that were present in our body before we were born that are present today. Right? So the point here is that these neurons are very, very long-lived, and they don't regenerate. They don't undergo mitosis and cytokinesis. In contrast, these neuroglial cells or glial cells do undergo mitosis and cytokinesis. These are incredibly abundant. They're super abundant. And again, there's 4 on the central nervous system. There's 2 in the peripheral nervous system. The functions in the peripheral nervous system include support and protection. Satellite cells and Schwann cells are the 2 examples in the peripheral nervous system. So we'll talk a little bit about those. So if you look at this picture right here, this picture on the left-hand side shows a single astrocyte, but it has a couple of different parts to it. Once again, now these astrocytes, this is the astrocyte with, these are the astrocytes, and this is the neuron. This is the neuron, I didn't have my glasses on. These are astrocytes, and these astrocytes have cell projections. Those cellular projections will go in 2 different directions here. In one direction, they attach to or cover the neuron, and then in the other direction, they go to the circulatory system, or the capillaries in this case. These processes that astrocytes have, called foot processes, surround both the soma and the capillaries, and they do a couple of things. The first thing that they do is that capillaries are incredibly leaky. Capillaries are built so that they're very, very small. They're not very thick structures at all. They're largely a single cell of epithelial tissue, and those unicellular squamous or simple squamous epithelial tissue is very thin and very flat, and so it allows lots of stuff to get out. That stuff that gets out, you don't want to get it, especially if it's in the brain. So the astrocytes, because this is what we're talking about here, astrocytes are in both the spinal cord and the brain. Astrocytes help build what's called the blood-brain barrier. The blood-brain barrier is pointing to what I'm right here. It helps promote junctions between cells, and those junctions between the capillary cells and the astrocytes themselves. And so it basically prevents substances that is injurious to the brain from getting out of the blood into the brain. Now, these astrocytes are not perfect because now when you guys are in about, let's just say you're 18 or almost 18, so in 3 years if you have a drink or two, alcohol can be a little disorienting. It can affect your brain. You guys haven't had any yet, so in the future when you do, Gianna, why are you laughing? So in the future when you do, what you'll find is you'll experience the alcohol does cross the blood-brain barrier. So these astrocytes are not perfect. Right? They don't prevent everything from crossing this blood-brain barrier, and there's an example of something that could be injurious. If at too great a concentration, it can be deadly. That is an example of the blood-brain barrier not functioning. So the general mechanism here is that the astrocytes are going to take nutrients, particularly glucose and oxygen, from the capillaries, from the plasma in the capillaries, and it's going to import it into the neuron itself, into the soma. So that gives the soma the ability to undergo high metabolic activity. Even when you're sleeping, your brain is operating. So that high and constant metabolic activity is occurring in the central nervous system. In addition, that metabolic activity is going to be getting rid of stuff. In particular, there's a number of different toxic waste material that you want to get rid of as the cell is developing, undergoing metabolism. There's a number of different acids, there's a number of different things that you want to get rid of. Carbon dioxide, another example. If you want to get rid of it, how do you get rid of it? You give it to the astrocyte, and then they put it back into the circulatory system. So the astrocytes they give everything that the neurons need, and they take anything the neuron doesn't want. That blood-brain barrier is an important critical element of the brain itself. So in other words, it's not found elsewhere in the body. And it also forms another layer of structure, another layer of covering on the brain, and that's called the pia mater. I haven't talked about the layer of coverings on the brain or on the spinal cord, but there's 3 layers of coverings on the brain or the spinal cord. There's a superficial, there's an intermediate, and then the deepest layer, the pia mater, is the layer by definition that touches the nervous tissue. So the combination of the pia mater covering the neuron, the astrocytes covering the neuron, those 2 things will help work together in supporting and protecting the neuron itself. The astrocytes also release chemicals to promote the development of synapses and things like that, and they help regulate synaptic activity. What does that mean? It helps keep the integrity of the synapses together, so that if you look on the left-hand side here, these are dendrites. The way this is drawn, these are dendrites, and there should be axons or axon terminals that are reaching over to get near to these dendrites. And so regulating and promoting the synaptic activity means that you want to keep these dendrites as close to the axons as possible, because you want to be able to have the information from the axons sent to those dendrites as quickly as possible. And that's helped largely by the astrocytes. Another of the neuroglial cells or glial cells are called ependymal cells. It's a weird word to say. Ependymal cells are areas found in spaces in the brain and the spinal cord. We haven't talked about what those are yet, but in a space in the spinal cord is called a central canal. The ependymal cells are also lining areas, spaces in the brain called ventricles. So your heart has ventricles and your brain does as well. These spaces then, the area, these areas that look similar to cuboidal epithelial cells, simple cuboidal epithelial cells, they have ciliary processes, and this drawing right here, these ciliated processes right here, they allow the ependymal cells produce cerebrospinal fluid. They take the plasma from the from blood, they modify the plasma from blood, and then they release through exocytosis the cerebrospinal fluid from the epidermal cells, and then the cilia then move the cerebrospinal fluid around. They try to circulate it, and in the process of circulating it, that cerebrospinal fluid helps move material throughout the brain and the spinal cord, and it helps nourish and protect and remove waste. There are a number of different areas in the brain. There are multiple areas in the brain where the cerebrospinal fluid is produced, and those are generally called choroid plexuses. That choroid plexus or choroid plexuses is found both in multiple places in the brain. Basically, every time there's a space in the brain called a ventricle, there's at least one or a group of choroid plexuses. A major cellular component of the cerebrospinal fluid is microglia. And those microglia are specialized macrophages, and so a macrophage is a big cell that undergoes phagocytosis. Anything that's crawling or swimming around in the cerebrospinal fluid that shouldn't be there is basically responded to by the microglia. They help phagocytize necrotic tissue. What does necrotic tissue mean?
Dead tissue.
So dead or damaged tissue, it helps do that. It helps in microorganisms and including foreign substances. Have you ever heard of college kids getting any neuronal diseases that are contagious? Anyone know of any of those?
You said neuronal?
Yep, nervous tissue diseases that are largely
College kids?
College kids.
College kids.
So it's
Senioritis? Senioritis?
senioritis?
Oh
No,
my
no,
gosh.
college, not high school kids.
Oh.
Didn't you say herpes? Didn't you say something about herpes?
So herpes is one example, right? But that's something that is, herpes will be, if it's damage in the skin, and then it damage into the neuron, right, the dendrite, and then through retrograde transport, it crawls back up into the axon. So that's one example, but this is not something that I've talked about. So there's levels of coverings in the brain, and those coverings, there's pia matter, arachnoid matter, and dura matter, and oh my God, I'm sleepy today because I forgot the name of it. Oh, I know. And those groups of layers that are covering the brain are called meninges, or spinal meninges, or cranial meninges. So meningitis is one example of a disease that's largely contagious, and lots of times college kids give it to each other because they're located so close together. There's two varieties of meningitis. Meningitis comes in either bacterial or viral. In both cases, if you get infected with it, and the first line of defense is going to be microglia, and the microglia will be the meningitis, these invading organisms, will be found in the cerebrospinal fluid, and the microglia will be fighting. If it overwhelms your microglia, then there's other medications that you can have. So one example of a foreign substance or a microorganism that is invading the cerebrospinal fluid would be result in meningitis. Another weird word here is oligodendrocyte, and oligodendrocytes are glial cells that produce myelin, and that myelin is the mechanism is important. So because the neurons are so closely together, they're in the central nervous system, the single oligodendrocyte has multiple cellular extensions, and those extensions then wrap around the axons particularly, and those axons then are wrapped by myelin. So oligodendrocytes make myelin. Oligodendrocytes wrap these myelin sheaths with by these cytoplasmic extensions that's branching off of the oligodendrocytes. And that single oligodendrocyte wraps multiple neurons. The area where the oligodendrocyte is wrapping the neuron is called the myelin, right here. And then the area where it's not wrapping it, that's the node. And in this case, there's a node of Ranvier. So now, if you see a picture where there's multiple axons that are wrapped by a single glial cell, that's got to be an oligodendrocyte. The mechanism that the oligodendrocyte produces is different than other mechanisms. In terms of what does myelin do, the first thing is that when in the brain, you can tell that these neurons are wrapped by myelin, because when the axons are wrapped by myelin, they appear to be white. So areas of the brain or the spinal cord that are white are, by definition, wrapped by oligodendrocytes and wrapped by myelin. Where are they wrapped? They're wrapped by the axons. And so that's the first thing. The second thing is that the oligodendrocytes then, and the myelin in particular, help do two things. The first thing is they help physically and chemically protect the axon from damage. So that if there's any pulling or prodding or anything like that, it's basically a wrapping of the neuron itself. And then the second thing is that it increases the velocity by what's called an order of magnitude. So if you're running at 10 miles an hour, if you then increase your speed of running by an order of magnitude, that means then you're running 100 miles an hour. Right? So that's how fast the action potentials then get sent in white matter versus gray matter. So it's very, very rapidly. And so the analogy that I like to draw here is that in this case, if this is a wire, right, and there's plastic or rubber that's wrapping it right there, that allows the signal to get sent from one area, from one end to the other when it's plugged in, and it protects it. It protects it, it supports it, it prevents any of the signal from dissipating. So that if this were plugged in and I grabbed it, would I get a shock? Well, if the integrity of it is intact, then I wouldn't get a shock. This is not entirely, this analogy is not entirely perfect, because there are these nodes. And these nodes, by definition, allow for the movement of ions into and out of the cell. And what are those ions? The sodium-potassium pump. So the action potential happens here, and then it happens here, and then it happens here, and that's called a particular type of action potential called saltatory action potentials. So oligodendrocytes will help increase action potential velocity here via this saltatory action potential production. So those are the four neuroglial cells that we'll talk about. Now we'll talk about just two in the peripheral nervous system. So the way that you should compare these, there's two that match up with in the central nervous system and the peripheral nervous system. One of them is Schwann cells, and a Schwann cell matches up with the oligodendrocytes. Schwann cells, there's many, many Schwann cells in our body, and those Schwann cells then a single Schwann cell wraps a portion of an axon. The way that happens is that the Schwann cell, through mitosis and cytokinesis, is produced. The Schwann cell encounters an axon, and then it starts producing myelin, and as it produces myelin, it wraps myelin in successively greater and wider areas. So a single neuron or a single neuroglial cell wraps myelin. So this is one neuroglial cell. This is another neuroglial cell. Here's a third neuroglial cell, and here's a fourth neuroglial cell. So these Schwann cells then they only cover a very small portion of the axon. These axons can be very, very long. The outer layer, the neurolemma, the way that I've seen it spelled, this book spells it with an I. The outer layer, I've seen it spelled with an O, the neurolemma. contains, is the neuron, the Schwann cell itself. So the neurolemma is the area of the Schwann cell that's wrapping the axon. And the protein is, again, myelin. In terms of when you're talking about whether it's Schwann cells or oligodendrocytes, the functions are exactly the same. The mechanism, the way in which these things are wrapping the axons are different, but the mechanism, the function is exactly the same. Physical and chemical protection, increase action potential velocity dramatically through saltatory action potential production. You see also in this diagram, so this area right here would be a part of a sensory neuron that is either a unipolar or a pseudo-unipolar neuron. Where my pointer is the axon, and that's labeled right there. And you can see right there where there's successive layers of the myelin that is produced by an individual Schwann cell. And then this is the major portion of the neuron itself, the soma or the cell body. And this soma or cell body is covered by the other neuroglial cell, and that's called a satellite cell. So a satellite cell is the area of the neuron or the neuroglial cell that surrounds the cell body or the soma. It's largely found in sensory ganglia and autonomic ganglia not because of anything in particular. That's where the cell bodies are found. What does it do? If you go back and you look at satellite cell, let me see here. These cells right here, astrocytes, and you look at the functions here, the functions of astrocytes and the functions of satellite cells are almost identical. They help cover the soma or the cell body. They provide nutrients to the soma or cell body. They physically protect it. The one thing that they don't do is they don't produce the blood-brain barrier because they're not in the brain, right? So they don't help that. The mechanism by which this happens here, providing nutrients, is different because the satellite cells don't have the cellular extensions that are present in the brain, and those satellite cells that don't have those extensions here, they wrap the soma or the cell body and they try to help provide the cell with nutrients. So I mentioned a little while ago the distinction between axons and those axons that are wrapped versus unwrapped. Those axons that are wrapped are called myelinated axons, and those myelinated axons generally are very, very long. And we talked about the structure of the myelin, regardless of where it's produced or how it's produced. The structure of the myelin will insulate and speed the axon, the action potential very, very rapidly. The area where the action potential will occur will be in a node, or the node of Ranvier. And when the myelin gets degraded, there's examples of myelin sheath that will degrade. There's a couple of examples. One example is in multiple sclerosis, and that's largely a neuromuscular disease. And another is example of diabetes. Diabetes in both cases, well, basically they cause a degeneration of the myelin sheath. When you hear about the term myelin, those axons are very long. They send signals very, very long distances. And in contrast here, gray matter are unmyelinated axons that are very, very short. We're going to say that they're not wrapped by the oligodendrocytes or Schwann cells. Unmyelinated axons are found solely in the central nervous system, whether it's in the spinal cord or the brain. And those gray matter axons then lack the material wrapping the axon, and they send action potentials in a continuous, slow way. So rapid saltatory action potentials are sent by white matter or myelinated axons. Slow, continuous action potentials are sent by unmyelinated or gray matter. Okay? And here is just, if you look at this top picture right here, here's a picture of myelinated axon, a single axon wrapped by two Schwann cells. I mentioned that nervous tissue does not regenerate. It doesn't undergo mitosis or cytokinesis, but that doesn't mean that it can't repair itself. Nerve cells are, neuronal cells are very, very long, and now I'm using this term nerves, right? So I'm talking about when a nerve is cut, right? And again, now that's multiple neurons that are cut along with the coverings associated with the neurons themselves. So this is nerves that are cut, part of the peripheral nervous system. What happens to them? First of all is not lost. Okay? If it's a severe cut, what would happen if you have a severe damage in the central nervous system that's in, let's say, the lumbar region that would cause dramatic, severe central nervous system damage? What consequence could that be to the person?
Paralysis?
What kind of paralysis?
Is it,
Would I be able to do this?
Yes.
would it be quadrant?
So when I said lumbar vertebrae. So where are lumbar vertebrae?
Or lower?
In the lower back. So would I be able to move my arms?
Yes.
So it all it depended on whether or not you get to the spinal nerves above the damage or below the damage. Spinal nerves below the damage will still be there, but they won't be functioning because they won't be sending signals. Well, then the signals wouldn't get to the brain, right, or to the muscles. So now, in the case that I just drawn through, the damage there that is occurred in the spinal cord, it would result in paralysis. But what kind of paralysis? Not quadriplegic, paraplegic, right? When you're talking about nervous tissue injury here, this is not this extent. Okay, you're talking about small numbers of nerves, perhaps a single nerve that's damaged, and the damage then can be responded to in this characteristic way. And so the distal axon breaks into segments, and that distal axon then dies. So wherever the damage occurs, distal to it, it dies. The Schwann cells that are near there, remember those Schwann cells are incredibly active, they start finding a new pathway for the remaining portion of the axon. And those Schwann cells then, with the help of macrophages as part of the immune system there, so basically some versions, so we're talking about leukocytes here now, they will invade those areas and help break up material that shouldn't be there, and the Schwann cells will enlarge, they divide, and they form a column so that it basically paves a new road for the axon to repair itself. When the axons then reattach, then you can have communication between the distal segment of the axon that was present, or that distal to where the damage was, and the regenerating area. Right? So now in the peripheral nervous system, you can have this repair, provided that it's small and it's not major. If you cut my arm off, right, and then you reattach it, the nerves are not going to find themselves back. Right? So we're not talking about something like that. We're talking about a minor damage. Regeneration in the central nervous system is limited. So the first part of that is just talking about regeneration in the peripheral nervous system. And its function, because largely the Schwann cells are helping. Oligodendrocytes basically prevent this because they're not able to provide new columns, or basically they don't find a new path for the axons to regenerate. So they're not allowing the axons to repair themselves, and so they prevent that. So oligodendrocytes don't help, Schwann cells do, but again, to a limited extent. Here is the picture again. Now this is either a neuron or a nerve, right? There's injury right there, distal to the injury, right here. It starts to atrophy. That atrophy, if it's not complete, then it can be repaired and it can reattach. If it is complete, then, right, then it's not gonna happen again. All right. So we talked a little bit about nervous tissue and nervous tissue repair. We talked about the structure of neurons and the function of neurons. We a little bit talked about the general terms for sending a signal or the action potential. We talked about the structure of the overall structure of the central nervous system and the peripheral nervous system, and the fact that gray matter found in the central nervous system is unmyelinated, and white matter, regardless of where it is, whether it's in the central or the peripheral nervous system, is myelinated. Those white matter areas there will send action potentials in a saltatory projection here. The gray matter sends signals in a action potentials in a continuous manner. Area of the brain, when we're talking about gray matter here, you're talking about the cortex of the brain is by definition gray matter. The areas of the brain, there are these, and I won't mention them right now, but there's areas that within the cerebellum, within the brain itself called the corpus callosum, there's particular areas of the brain that's white matter that's surrounded by gray matter. In the central nervous system, when you, as opposed to in the peripheral nervous system, when you group neuronal cell bodies together, you call them nuclei. So now we have a cell nucleus, we have the atom of a nucleus, and now we have a nucleus in the brain. The nucleus in the brain is very, very different than the other two nuclei. And so that's just bundles of cell bodies that are found within the central nervous system. And nerve tracts there are essentially myelinated axons that are found within the central nervous system. We talked about ganglia and nerves already. Nerves are part of the peripheral nervous system. Ganglia are part of the peripheral nervous system. Ganglia are these cell bodies there. All right, so we're gonna stop right here. The vocabulary that I've used today is pretty significant. And what we're gonna start doing tomorrow and then on Monday is talk about two major topics, all the things that happen at a synapse and all the major things that happen during an action potential. And those get into really complex terminology.